Hello, my dear students. Welcome to Takshar Academy. So, in the previous classes, we have discussed about the some important concept of the biotechnology and related to the process and principle and different types of tools, which is very much necessary for the formation of the recombinant molecules. That is a different variety of product with a variety of steps will be involved in the biotechnology with a process and principle. And in the next chapter of the biotechnology concept, that is biotechnology and its applications. We are well known the biotechnological applications are in the field of variety types of industry. For example, if you take a medicinal field or if you take a agriculture field or if you take any kind of preparation of different variety of the product, we can definitely go for the biotechnology. And we have to extract the maximum number of product, we can go for only a biotechnology. So that in the chapter of biotechnology and its applications, we have studied about the major content, the content number one, that is applications related to the agriculture field and applications related to the medicinal field and the technique of gene therapy and the technique of molecular diagnosis and the mechanism of one important process, the production of human insulin artificially and the development of the pest along with the insect resistance plant. And also we have studied about transgenic animals also and their applications of transgenic animals. Okay. And today's class, we are discussing about the some questions which is related to the concept of biotechnology and its application procedure. Okay. So we're coming to the question number one. The study of the following statement regarding the Bt toxin produced by a bacteria, that is a Bacillus thuringiensis, and you have to select the correct options for the following. So, four statements they are given. The related to the concept of that is Bacillus thuringiensis. We have to insert it into the cotton plant. So we can call Bt cotton. So related to Bacillus thuringiensis, what is the major functions of Bacillus thuringiensis gene? What is the function and what is the mechanism of the Bacillus thuringiensis? So according to the concept of this, the statement number one, the first one, the some kind of strain that is totally related to the Bacillus thuringiensis, they have to production of the protein that can ability to kill the certain type of insect such as a lepidopteran and there is a coleopterans and if you take an example for the dipterans. So ಇಲ್ಲಿ example of the strain of related to the Bacillus thuringiensis ಅಂತ ತಗೊಂಡ್ರೆ they are very important protein will be producers and that protein it is a majorly it can kill the certain type of insect, that is we can call the lepidopterans and coleopterans along with a different variety of dipterans also. So these protein it is a very important and it can be rupture the gut wall of insect all the type of organism which are consumed by the like the some kind of example of Betty cotton or different types of organism. So that is we can call insecticidal protein. Okay, ಆ ಒಂದು ಪ್ರೋಟೀನ್ ನಾವೇನಂತ ಕರಿತೀವಿ ಇನ್ಸೆಕ್ಟಿಸೈಡಲ್ ಪ್ರೋಟೀನ್ ಅಂತ ಕರಿತೀವಿ. So it can kill the insect. So it is a statement number correct. And if you take a second one, the Bt toxin protein do not kill the bacteria themselves because the toxin production occurs in an inactive form, that is we can call the protoxin. So ಇಲ್ಲಿ the Bt toxin protein ಏನಿದೆ and it will not kill the specific bacteria themselves. A ಬ್ಯಾಕ್ಟೀರಿಯಾದಿಂದನೇ ಪ್ರೊಡಕ್ಷನ್ ಆಗ್ತಕ್ಕಂಥ ಪ್ರೋಟೀನ್ it will not affect for the bacteria because of what is the reason behind that ಅಂತ ತಗೊಂಡ್ರೆ that is a majorly the production of protein from the bacteria it is a completely inactive form. ಸೊ ಆ ಒಂದು ಪ್ರೋಟೀನ್ ಏನಿದೆ ಆಲ್ವೇಸ್ ಹೆಂಗಿರುತ್ತೆ ನಮಗೆ inactive form ಅಲ್ಲಿ ಇರುತ್ತೆ. Okay, and that inactive protein we can consider the formation of a protoxin ಅಂತ ಕರಿತೀವಿ. And these protoxin it can interact with a alkaline media. ಸೊ ಅಲ್ಕಲೈನ್ ಮೀಡಿಯಾ ಜೊತೆಗೆ ಈ ಪ್ರೋಟಾಕ್ಸಿನ್ ಏನಾದ್ರೂ ಇಂಟ್ರಾಕ್ಟ್ ಆದ್ರೆ ಇಟ್ ಕ್ಯಾನ್ ಬಿ ಕನ್ವರ್ಷನ್ ಇಂಟು ಏನಾಗಿ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ ಅಂದ್ರೆ ದಟ್ ಈಸ್ ಕನ್ವರ್ಷನ್ ಆಫ್ ಟಾಕ್ಸಿನ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ. ಸೊ ದಟ್ ಸ್ಟೇಟ್ಮೆಂಟ್ ನಂಬರ್ 2 ರೈಟ್. ಅಂಡ್ ಸ್ಟೇಟ್ಮೆಂಟ್ 3 ನೋಡ್ಕೊಳ್ಳಿ. ವೆನ್ ಆನ್ ಇನ್ಸೆಕ್ಟ್ like ingest the inactive bt toxin and it is completely converted into active form due to alkaline pH. We are just studied that is a alkaline pH is very very important, and these alkaline pH of the gut of the insect which is completely solubilize the protein that is a crystal form of a protoxin converted into a the liquid form of a toxin, and it can completely affect for the insect and the death of an insect will be occurs. So it is also be a statement number 3 the correct one. So according to the question number 1 the statement related to the bacillus thuringiensis bacterium, so that is the all of the above will be the correct option. Okay, next we'll coming to the question number 2. The bt toxin gene has been cloned from the bacteria and they have to express it in the plant production of resistance to the insect without the need of insecticides. And give an example for such a plants ಅಂತ ಕೇಳಿದಾನೆ. ಸೊ ಇಲ್ಲಿ bt toxin ಅನ್ನೋದೇನು, it is a some kind of gene has been cloned from the bacteria and they have to express it in the plant. ಆ ಬಂದಿರ್ತಕ್ಕಂಥ bt toxin gene ಏನಿದೆ they have to express it in the plant and plant can get some kind of resistance either insect or any kind of different worm. So without usage of any kind of insecticides ಅಂತ ಕರಿತೀವಿ. And example for that plants ಅಂತ ಕೇಳಿದಾನೆ. So that plants are variety of types of plants. If you take an example for the cotton and if you take example for the corn, you have to insert the bt toxin gene. And if you take a rice also and potato also tomato also and if you take any kind of soybean and all the plant species we have to insert it directly, that is a bt gene, that is a bt toxin gene, so that all the options will be correct. So that question number 2 the correct option should be the option D will be the correct one because of example for the modified variety, that is a genetically modified organism which is comes under the plant. The bt cotton will be available, bt corn also, bt rice belong with a bt tomato, potato and soybean. All the wide variety of the modified plant will be available, that is a genetically modified organism. So the question number 2 the correct option should be the option D. The next question number 3. Which of the following statement regarding the structure of proinsulin and mature insulin are not correct? So ಇಲ್ಲಿ very very important point idu. So the applications which is related to the production of a human insulin in the artificial manner. So ಇಲ್ಲಿ ಕೊಟ್ಟಿದ್ದಾನೆ ವರ್ಡ್ that is a structure related to the proinsulin and you have to differentiate the mature insulin ಅಂತ ಕೊಟ್ಟಿದ್ದಾನೆ and which one is not correct ಅಂತ find out ಮಾಡಬೇಕು. So if you take the before going to the question, we are well known the insulin it is totally produced in ಎಲ್ಲಿ ಪ್ರೊಡಕ್ಷನ್ ಆಗುತ್ತೆ ಅಂದ್ರೆ ವಿತ್ ಇನ್ ದಿ ಪ್ಯಾಂಕ್ರಿಯಾಸ್. ಸೊ ಪ್ರೊಡಕ್ಷನ್ ಆಗ್ತಕ್ಕಂಥ ಸೈಟ್ ಎಲ್ಲಿ ನಮಗೆ ಪ್ಯಾಂಕ್ರಿಯಾಸ್ ಅಲ್ಲಿ. ಸೊ ವಿ ಆರ್ ವೆಲ್ ನೋನ್ ಆಲ್ಫಾ ಸೆಲ್ ಅಲಾಂಗ್ ವಿತ್ ಎ ಬೀಟಾ ಸೆಲ್ಸ್ ಅನ್ನೋದು. And the beta cells immediately basically they have to involved in the production of a human insulin and they have to regulate the glucose level. Okay, but ಇಲ್ಲಿ artificially production of human insulin ಅಂತ ತಗೊಂಡಾಗ first the artificially we have to extract the insulin from the dog. That is a ಡಾಂಗ್ ಪ್ಯಾಂಕ್ರಿಯಾಟಿಕ್ ಇಂದ ನಾವು ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಮಾಡ್ತೀವಿ ಅಂಡ್ ಪಿಗ್ ಪ್ಯಾಂಕ್ರಿಯಾಟಿಕ್ ಇಂದನು we have to extract it. Cattle ಇಂದನು we have to extract the insulin. But all the type of the organism production of insulin, that insulin is injected into the person and that person shows some kind of abnormal symptoms or abnormal allergic reactions will be occurs, so that it can be totally avoided. ಸೊ ಈ ನಾವು ಅನಿಮಲ್ಸ್ ಇಂದ ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಮಾಡ್ತಕ್ಕಂತ ಇನ್ಸುಲಿನ್ ನಾವೇನ್ ಮಾಡಿದೀವಿ, ಸ್ಟಾಪ್ ಮಾಡಿದ್ವಿ, ಬಿಕಾಸ್ ಆಫ್ ಇಟ್ ಕ್ಯಾನ್ ಶೋ ಅಡ್ವರ್ಸ್ ಎಫೆಕ್ಟ್. ಓಕೆ, ಬಟ್ ಈಗ ಆರ್ಟಿಫಿಷಿಯಲಿ ನಾವು ಪ್ರೊಡಕ್ಷನ್ ಮಾಡಬೇಕು ಅಂದ್ರೆ that is we can go for the one of the important technique, that is RDNA. ಸೊ ಹ್ಯೂಮನ್ ಇನ್ಸುಲಿನ್ ಪ್ರೊಡಕ್ಷನ್ ಮಾಡ್ತಕ್ಕಂಥ ಟೆಕ್ನಿಕ್ ಯಾವ್ದು ನಮ್ಗೆ ಇಲ್ಲಿ ಎಕ್ಸಾಂಪಲ್ ತಗೊಂಡಿದ್ದೀನಿ, ದಟ್ ಈಸ್ ಎ ಮೆಕಾನಿಸಮ್ ಆಫ್ RDNA. ಏನಿದು RDNA ಅಂದ್ರೆ ರೀಕಾಂಬಿನೆಂಟ್ DNA ಟೆಕ್ನಾಲಜಿ. And we have to extract the gene which is a production of insulin within the BT, like example of beta cell of a pancreas, islets of Langerhans of beta cells. We have to extract the gene. Artificially, we have to clone it in a bacteria and we have to prepare the different chain of insulin and we have to get an artificial insulin. So that is we can consider the pro insulin is totally different from the mature human insulin anta kare. So ಇಲ್ಲಿ the pro insulin is nothing but it is a directly production within the human being. Human beings in the production ಆಗ್ತಕ್ಕಂಥ ಇನ್ಸುಲಿನ್ ಹೆಂಗಿರುತ್ತೆ, pro insulin, that is we can call inactive. And normally insulin ಅಂತ ತಗೊಂಡಾಗ we can go for the technique of RDNA. ಯಾಕೆ ಇದು ಪ್ರೋ ಇನ್ಸುಲಿನ್ಗೂ ಒಂದು ಮೆಚೂರ್ ಇನ್ಸುಲಿನ್ಗೂ ಏನು ಡಿಫರೆನ್ಸಸ್ ಇದೆ ಅಂತಂದ್ರೆ, the pro insulin it is directly produced from the human insulin and that human insulin it can be converted into mature insulin ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ. ಓಕೆ, ಬಟ್ ಇಲ್ಲಿ ಪ್ರೋ ಇನ್ಸುಲಿನ್ ಇಸ್ ಟೋಟಲಿ ಮೇಡ್ ಅಪ್ ಆಫ್ ಎಷ್ಟಿರುತ್ತೆ ನಮಗೆ ಪಾಲಿಪೆಪ್ಟೈಡ್ ಚೈನ್ಸ್ ಅಂತ ತಗೊಂಡ್ರೆ 3 ಚೈನ್ಸ್ ಬಿ ಅವೈಲೇಬಲ್. ಎಕ್ಸಾಂಪಲ್ ನಂಬರ್ 1 A, B and C. A chain, B chain and C chain we can consider the formation of the pro insulin. Okay, and statement 1 will be a correct. And if we take a second statement, C polypeptide chain with 33 amino acids. So ಇಲ್ಲಿ ನೋಡು 3 like chains will be present, and out of 3 if you have taken example for the C polypeptide chain ಅಂತ ತಗೊಂಡ್ರೆ they have made up of a 33 amino acid is removed prior to the human insulin formation ಅಂತ ಕೊಟ್ಟಿದ್ದಾನೆ. So before going to the formation of, so formation conditions ಅಲ್ಲಿ ನೋಡೋದಾದ್ರೆ ನಾವು, so 33 amino acids will be present and you have to remove before the formation of human insulin, that is a mature, so that it is a correct one. So ಈಗ ನಮಗೆ ಏನು ಗೊತ್ತಾಯ್ತು, completely they have made up of a 3 different chain, that is a chain will be present, B chain, C chain, and C chain made up of a 33 amino acid. Okay, so that option B correct one. And if you take a third one, the mature human insulin is made up of totally 51 amino acids. Definitely they have only 51 amino acids will be present out of 51. So out of 51 ಅಂತ ಬಂದಾಗ ನಮಗೆ 51 ಯಾವ್ದು ಯಾವ್ದು ಇರುತ್ತೆ ಅನ್ನೋದು ಕೂಡ ವೆರಿ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ಸೊ ಇಲ್ಲಿ the A chain ಅನ್ನೋದು ಏನಿದೆಯಲ್ಲ that is a 21 and B chain ಅಂತಕ್ಕಂಥದ್ದು ಏನಿರುತ್ತೆ 30 ಇರುತ್ತೆ. ಸೊ ಎರಡನ್ನು ಕಾಂಬಿನೇಷನ್ ಮಾಡಿದ್ರೆ we can get ಎಷ್ಟು ಬರುತ್ತೆ ನಮಗೆ ಫಾರ್ಮೇಶನ್, 51 amino acids will be present. So statement number 3, arranged in a 2 polypeptide chain with a disulfide bridges and A and B chain will be, it is also be correct one. But we have to find out the not correct options according to the pro and human mature insulin. And next statement, the polypeptide chain A has 30 amino acid and polypeptide chain B has a 21 amino acid. So ಇಲ್ಲಿ A chain is completely made up of a 21 and B chain is made up of a 30. So that option number D, wrong statement. And the polypeptide chain of A and B are interconnected by only a one sulfide bridge ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. Many sulfide bridge will be required only, it is not correct. So that statement 4th and statement 5th will be totally wrong and not correct related to the human insulin and pro insulin. So that option ಅಲ್ಲಿ the C will be the correct one. 4 and 5th will be totally not correct. Okay na. And ಇಲ್ಲಿ meaning is that let us consider this is the human insulin and A chain will be present and B chain will be present, C chain. But when going to the maturation of the pro-insulin, that is considered the pro-insulin. Okay, insulin will be pro. And we'll coming to the maturation of insulin anta bandaga A chain interact with a B chain only. So that A and B interacted with a disulfide bridges only. And totally the removal of C chain, that is we can consider the pro-insulin converted into a well-known example of human insulin. That is we can call it as a example of formation of a related to the maturation of insulin. So optionally ನಮಗೆ C will be the correct option for the question number 3. Next question number 4. Read the given statement and you have to select the correct options. Statement number 1. TI plasmid, tumor inducing plasmid obtained from Agrobacterium tumefaciens and is effectively used as a vector for gene transfer in the plant cells. And statement 2 ನೋಡ್ಕೊಳ್ಳಿ the part of TI plasmid transformed into the DNA of the plant cells and is called the tDNA. So according to the statement wise and which one is a true and false and explanation related questions ಇದು. So ಇಲ್ಲಿ statement number 1 ನೋಡ್ಕೊಳ್ಳಿ the TI plasmid obtained from Agrobacterium tumefaciense. So this is the one kind of a organism nematode anta ಕರಿತೀವಿ and these nematode they have capability to produce some kind of a the gene which is a very used for the induction of tumor. The tumor will be totally induced, that is TI plasmid we have to extracted or obtained from the Agrobacterium tumefaciense effectively used as a vector for the gene of a transferring to the plant. ಇದು carrier ಆಗಿ act ಆಗುತ್ತೆ and it can induce the growth and development of a tumor. So statement number 1 correct. And part of TN, like example of TI plasmid, transformed into the DNA of the plant cells and is called the tDNA. So ಈ TI plasmid in transport ಮಾಡ್ತಾ ಇದೆ gene of DNA ಅಥವಾ set of sequence of DNA. That DNA we can consider the tDNA ಅಂತ ಕರಿತೀವಿ. tDNA ಅಂದ್ರೆ ಮೀನಿಂಗ್ ಟ್ಯೂಮರ್ ಇಂಡ್ಯೂಸಿಂಗ್ DNA ಅಂತ ಕರಿಬಹುದು. ಏನ ಟ್ರಾನ್ಸ್ಫರ್ DNA ಅಥವಾ tDNA ಅಂತಾನೂ ಕರಿತೀವಿ. So both the statement will be true. But related to the explanation anta bandaga statement number 1, both statement 1 and 2 will be the correct and statement 2 is a correct explanation regarding the statement 1 anta ide. It is not explanation related, so that statement 1 and 2 correct but not correct explanation. Next, both this statement will be correct but this statement 2 not the correct explanation for the statement 1, so it is a true. And statement 1 is correct, 2 is incorrect anta ide and both will be incorrect anta ide, so ಯಾವುದು ಬರಲ್ಲ. So option ಅಲ್ಲಿ the correct option should be the option B will be the correct one for the question number 4. Okay, next the question number 5. Like example the green evolution. So based on the green revolution has been the resounding the success in terms of agriculture production and it has failed in its overall social objectives. Because of what is the reason behind that the failure of green revolution all over the country. So overall social objectives ಅಂತ ತಗೊಂಡಾಗ it has failed. ಯಾಕೆ ಈ green revolution ಅನ್ನೋದು completely they are related to the field of agriculture production in the simultaneous way with respect to the complete production in a same way, and it is a failed in a specific area with related to the particular country. Why there is a failure of green revolution? ಯಾಕೆ ಇದು ಫೇಲ್ಯೂರ್ ಆಗಿರ್ತಕ್ಕಂಥ ಪ್ರಾಜೆಕ್ಟ್ ಅಂತ ತಗೊಂಡ್ರೆ because the reason ಏನಿದ್ರೆ it has not succeeded in making India totally and permanently self-sufficient in food. With a green revolution in the total permanent method irbodu and self-sufficient in the food ಅನ್ನೋ ಒಂದು process ಗೆ that is a succeeding making really totally they are failed in an India, so that it is a failure of objectives ಅಂತ ತಗೊಳ್ತೀವಿ. So statement 1 is correct. And use of agrochemicals ಏನಿದೆ become very very expensive for the Indian peoples or farmers, as well as these have the harmful effect for the environment. So ಈ green revolution conduct ಮಾಡ್ಬೇಕಂದ್ರೆ we have to use some kind of agro based chemical. We can buy the different types of insecticides and pesticides to control the pest and insect and overgrowth and over yield increase. But over utilization of these like example of chemicals, they are highly affect the environment because of it can create a pollution and it can damage the ecosystem and it can completely eradicate the some kind of micro and macro habitat which is already present on the soil. So it is also one of the important and they are very expensive. It is a very very most important point. If you take any kind of agro based chemicals, they are highly expensive, so that the poor farmers will not buy this kind of agro based chemicals, so that it is totally failure of green revolution. So optionally B also be right. And if you take a C, in example of regional terms, only a Punjab with respect to the Haryana states and the some kind of eastern plains of a river Ganga and West Bengal state showed the reasonable good result, but results were less impressive in an other part of an India because of availability of the water is more sufficient and it can show the good result. But if we take a different other parts of the other state in an India, the availability of the water, the scarcity of water, and availability of expansive nature of agro-based techniques and a wide variety of the agriculture field technique, they are not known. So that all the terms and conditions with related to the some kind of social objectives, the completely the green revolution concept will be filled in an India. So the optionally ನೋಡೋದಾದ್ರೆ all the above will be the correct one. So option C will be option D will be the correct for the question number 5. And if you take a question number 6, which of the following statement regarding the gene therapy is are correct? So if we take an example for the gene therapy, we are well known meaning of gene therapy. Gene therapy ಅಂದ್ರೆ meaning ಏನು ಕರಿಬಹುದು ನಾವು? It is a replacement of a defective gene with the insertion of healthy or the modification of a gene, defective gene we are modified. Okay. And we can go for the formation of a preparation of a new combination or a new kind of organism. That is we can call the gene therapy technique. Okay. So ಇಲ್ಲಿ with a well important treatment already we are succeeded or they can get a success in the field of gene therapy with a curing of like deficiency syndrome of ADA gene also. We are well known that. Okay. And according to the which of the statement regarding the gene therapy, it could be right and the Togondaga. And it is an applications of biotechnology. Definitely, it is one of the application related to the biotechnology. And in which the defective gene is manipulated introduction of a normal. Or we have to take an example of healthy or functional gene. So in the gene therapy ಅನ್ನೋದೇನು, it is a one important biotechnology and it can be modification. Defective gene is completely modified into a normal healthy gene or a functional gene. That is we can call the biotechnology related to the gene therapy statement. One definitely correct. And two, the genetic disorders that are being investigated for the gene therapy range from the sickle cell anemia and severe combined immunodeficiency syndrome or the deficiency disease. So with a gene therapy technique, we have to investigate the variety of the different diseases with a modification. And one of the most important examples they are given, sickle cell anemia, the shape of the RBC they have to completely constricted because of the deficiency of or the modification of arrangement of the amino acid sequence or a coding property. And already we are well known about the CAD, that is nothing but a severe combined immunodeficiency, and it is totally treat regarding the treatment for the ADA deficiency syndrome. That is also the correct option C. And first, the clinical gene therapy was given in the year of 1990s with a 4-year-old girl with a deficiency syndrome. That is we can call the adenosine deaminase deficiency property. So that option C also be correct one. And according to the statement related to the question of the gene therapy, all the statements should be the correct option. D will be the correct option for the question number 6. Okay. The next question number 7. Which of the following statement is correct with regard to the disadvantages of genetically modified crops? And with the insertion of genes with respect to the specific plant, we have to get a different types of advantages. They are resistance, they are more yield, they have high nutrition, okay, and they are healthy, and they are specific, and they are resist toward the different pest along with the insect, disease free plant. So these are many types of advantages which is related to the genetically modified crops. But while coming to the statement of disadvantages regarding that, the genetically modified crops can be like completely these crops can be affect human by causing allergic reaction. Statement number one. If we take the genetically modified crops, it can affect to the human and it can be completely give some kind of allergic reactions with a overutilization of these crops ಅಂತ ಕರಿತೀವಿ. So that statement number A, it is a correct one. And transients in an commercially, that is we have to take example of crops can be endangered, and like example of native species, and the bt toxin gene is expressed in the pollen might have endangers the pollinators, like example insects. ಈಗ ನಾವು ಟ್ರಾನ್ಸನಿಕ್ ಅನಿಮಲ್ಸ್ ಅಂತ ಏನ್ ಕರಿತೀವಿ, ಲೈಕ್ ಎಕ್ಸಾಂಪಲ್ ಆಫ್ ಟ್ರಾನ್ಸನಿಕ್ ಕ್ರಾಪ್ಸ್ ಅಂತ ನೋಡಿದಾಗ ಮಾಡಿಫೈಡ್ ಜೀನ್ಸ್ ಇರ್ತಕ್ಕಂಥ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ with the insertion of foreign DNA or the modification of gene, they have to involve the some kind of killing activity of pollinators. Example, variety of insect. ಇಲ್ಲಿ ಇನ್ಸೆಕ್ಟ್ ಏನಾದ್ರೂ ಪ್ರೆಸೆನ್ಸ್ ಇಲ್ಲ ಅಂತಂದ್ರೆ there is no pollination, and without pollination there is no production of a new seed or a new fruit. But these genetically modified crops have affected to the some kind of pollinators. Example, honeybees irbodu, insects irbodu, different types of examples effect ಕೊಡ್ತಕ್ಕಂಥ. Okay, so statement 2 also be correct one. And C, the production of genetically modified crops causes the damage to the natural environment and it's always costly. So again now genetically modified crops ಅಂತ ಕರಿತೀವಿ, they are damage to the natural environment. The natural environment nothing but a killing of different types of a habitat related to the microorganism or any kind of bacteria, so that it can be affect to the natural environment one point, and it is always a costly nature. So in the process in it is genetically modified organisms, so it is a very very costly procedure. Okay, so statement number related to the seventh ಅಂತ ತಗೊಂಡಾಗ disadvantages, all they will be the disadvantages, so that the correct option should be the option D will be the correct one for the question number 7. The next question number 8. The following figures, it can be show the different steps which are involved in the production of genetically engineering insulin, and you have to identify the enzyme involved in the steps A and B and C, and you have to select the correct statement regarding the point. So ಇಲ್ಲಿ ಒಂದು ಇಂಪಾರ್ಟೆಂಟ್ ಎಕ್ಸಾಂಪಲ್ಸ್ ಕೊಟ್ಟಿದ್ದಾರೆ, ಹೌ ಯು ಹ್ಯಾವ್ ಟು ಪ್ರೊಡಕ್ಷನ್ ಆಫ್ ಲೈಕ್ ಎಕ್ಸಾಂಪಲ್ ಆಫ್ ಇನ್ಸುಲಿನ್, ನಾವು ಯಾವ ತರ ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಮಾಡ್ತೀವಿ, ಯಾವ ತರ ಇನ್ಸರ್ಟ್ ಮಾಡ್ತಾ ಇದೀವಿ, ಪ್ರಿಪರೇಷನ್ ಆಫ್ ಆರ್ಟಿಫಿಷಿಯಲ್ ಹ್ಯೂಮನ್ ಇನ್ಸುಲಿನ್, different steps with a graphical representation and diagrammatic representation they are given. So you have to find out the specific type of marking. A anta marking ide, so you have to observe the diagram alli ನೀವು observe ಮಾಡ್ತಾ ಹೋದ್ರೆ ಇಲ್ಲಿ A ಅನ್ನೋ marking ಸಿಗುತ್ತೆ, C ಇದೆ, and if you take a B also, ABC marking system will be available. And regarding the ABC marking system, and you have to find out the which one will be the correct for the option A and B and C. So ಇಲ್ಲಿ ನೋಡಬಹುದು ನಾವು. So first if you take an example like question number 8, the enzymes involved in the steps of A and B are the same, that is a restriction exonuclease ಅಂತ ಇದೆ. ಸೊ ನಾನು RDNA ಟೆಕ್ನಿಕ್ ನ ಕಂಡಕ್ಟ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ, ರಿಕಾಂಬಿನೆಂಟ್ DNA ಪ್ರೊಡಕ್ಷನ್ ಆಗ್ತಾ ಇದೆ ಅಂದ್ರೆ ವಿ ಕ್ಯಾನ್ ಗೋ ಫಾರ್ ದಿ ಡೆಫಿನೆಟ್ಲಿ ಎಕ್ಸೋ ಹೋಗಲ್ಲ. ನಾನು ತಗೊಳ್ತಕ್ಕಂತ ಎಕ್ಸಾಂಪಲ್ಸ್ ಏನಿರ್ಬೇಕು, ನನಗೆ ಎಂಡೋ ನ್ಯೂಕ್ಲಿಯಸ್ ಎನ್ಸೈಮ್ಸ್ ಇರ್ಬೇಕು. So option A, the enzymes which is involved in these steps A and B and are same, that is a restriction endonuclease ಅನ್ನೋದ್ರೆ ರೈಟ್ ಆಗುತ್ತೆ, exonuclease will not correct. So that option wrong. And we'll coming to the B statement ನೋಡ್ಕೊಳ್ಳಿ ಇಲ್ಲಿ. The enzymes which is involved in this step B and C are same. B and C ಅಂತ ಕೊಟ್ಟಿದ್ದೇನೆ, B and C will be the same. And restriction endonuclease enzymes ಅಂತ ಇದೆ. So ಇಲ್ಲಿ B and C will be the same ಅಂತ ಬಂದಾಗ ವರ್ಡ್, ಜಸ್ಟ್ ಯು ಹ್ಯಾವ್ ಟು ಅಬ್ಸರ್ವ್ ದಿ ಡಯಾಗ್ರಾಮ್. ಸೊ ನೀವು ಡಯಾಗ್ರಾಮ್ ನ ಅಬ್ಸರ್ವ್ ಮಾಡಿದ್ರೆ ಏನ್ ಗೊತ್ತಾಗ್ತಾ ಇರೋದು, ಈ ಒಂದು ಬಿ ಅನ್ನೋ ಸ್ಟೆಪ್ ಅಲ್ಲಿ ದ ಪ್ಲಾಸ್ಮಿಡ್ ಕ್ಯಾನ್ ಬಿ ಕಟ್. ಪ್ಲಾಸ್ಮಿಡ್ ಏನಾಗಿದೆ, ಕಟ್ ಆಗ್ತಾ ಇದೆ. ಸೊ ಪ್ಲಾಸ್ಮಿಡ್ ಕಟ್ ಆಗಿದೆ ಅಂದ್ರೆ ದೇ ಆರ್ ಕಟ್ ಓಪನ್, ಸ್ಟ್ರೆಚಸ್ ವಿಲ್ ಬಿ ಪ್ರೆಸೆಂಟ್. ಅಂಡ್ ಸಿ ಅಲ್ಲಿ ನೋಡಿದ್ರೆ ಅಗೇನ್ ದೇರ್ ಇಸ್ ಅ ರೀಸೀಲಿಂಗ್ ವಿಲ್ ಬಿ ಕಂಡಕ್ಟೆಡ್. ಸೊ ಇಲ್ಲಿ ಸಿ ಅನ್ನೋದೇನು, ಇನ್ಕಾರ್ಪೊರೇಷನ್ ಆಫ್ ದ ಫಾರಿನ್ DNA ಇಂಟು ದಿ ಪ್ಲಾಸ್ಮಿಡ್ ವಿತ್ ಯೂಸ್ ಆಫ್ ಅನ್ ಎನ್ಸೈಮ್. ಸೊ ಯಾವ ಎನ್ಸೈಮ್ ಯೂಸ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ ಅಂದ್ರೆ ನಾನು ಅಲ್ಲಿ DNA ಲೈಗೇಸ್. ಸೊ ಎನ್ಸೈಮ್ ನೇಮ್ ಯಾವ್ದು ನಾನು ಯೂಸ್ ಮಾಡ್ತಾ ಇರೋದು, DNA ಲೈಗೇಸಸ್. ಅಂಡ್ ಇಲ್ಲಿ ಸೇಮ್ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸ್ ಅಂತ ಕೊಟ್ಟಿದ್ದಾನೆ, ಸ್ಟೇಟ್ಮೆಂಟ್ ನಂಬರ್ 2 ಫಾಲ್ಸ್. ಅಂಡ್ ನೆಕ್ಸ್ಟ್ ಸಿ ನೋಡ್ಕೊಳ್ಳಿ, ಎನ್ಸೈಮ್ಸ್ ಇನ್ವಾಲ್ವ್ಡ್ ಇನ್ ದಿ ಸ್ಟೆಪ್ಸ್ ಆಫ್ ಸಿ ಆರ್ DNA ligase. Only they are given C steps. T step is indicated for the incorporation of the foreign DNA into the plasmid, so that there is a ligated and automatically the DNA ligase is a correct option. Okay. And the next, both A and C will be the anta kottidane, it is a totally wrong. According to the diagrammatic representation, the statement C will be the correct one. So ಇಲ್ಲಿ A ಅಂದ್ರೆ ಮೀನಿಂಗ್ ಏನಾಗುತ್ತೆ ನಮ್ಗೆ, use of a endonuclease for the extraction of DNA, cutting a plasmid with the use of a endonuclease, and both the endonuclease part or with respect to the DNA segment we are to join with the use of a C, that is nothing but a DNA ligase enzyme. So that according to the ABC statement anta nododadre, the correct statement should be the option C will be the correct one for the question number 8, the DNA ligase affect C chain. Okay. Next, the question number 9. Read the given statement and you have to select the correct options anta kottidane. And regarding the statement number 1, the recombinant DNA technology, human genes are often transformed into bacteria, that is an example of prokaryotes, or there is a yeast, that is an example for the eukaryotes. ಇಲ್ಲಿ ಎರಡು ತರ ನಾನು ಯೂಸ್ ಮಾಡ್ತಾ ಇರೋದು, ದಟ್ ಇಸ್ ರಿಕಾಂಬಿನೆಂಟ್ DNA ಅಂತ ತಗೊಂಡಾಗ ಹ್ಯೂಮನ್ ಜೀನ್ಸ್ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡ್ತಾ ಇರೋದು ಪ್ರೋಕ್ಯಾರಿಯೋಟ್ ಇರ್ಬೋದು ಅಥವಾ ಯೂಕ್ಯಾರಿಯೋಟ್. ಯೂಕ್ಯಾರಿಯೋಟ್ ದಲ್ಲಿ ಸ್ಪೆಸಿಫಿಕ್ ಎಕ್ಸಾಂಪಲ್ ಇಷ್ಟು, ಆಲ್ಮೋಸ್ಟ್ ಸ್ಪೆಸಿಫಿಕ್ ಇನ್ ಎ ಪ್ರೋಕ್ಯಾರಿಯೋಟ್ ದಲ್ಲಿ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ಓಕೆ. And both the bacteria and yeast, they have to multiply very fast and to form the huge population and which can express the desired gene. So ಇದು ಕ್ರಿಯೇಟ್ ಆಗ್ತಕ್ಕಂಥ ಪಾಪ್ಯುಲೇಷನ್ ಏನಿರುತ್ತೆ, ವೆರಿ ಮ್ಯಾಕ್ಸಿಮಮ್, and it is a very fast also, and they have to express and they have to desired genes will be present and they have to production, so that both the statement will be correct. And correct explanation regarding the statement 1, so that both the statement will be 1 and 2 will be the correct, so that option, and 2 is the explanation for the A statement, it is also correct one, so that option A will be the correct one for the question number 9. Because of prokaryote and eukaryote, and we have to take a multiple copy with a fraction of or the limited time, and desired gene also we have to extract, so that option B, A will be the correct. The next question number 10. The study the following statement and you have to select the incorrect ones. So they are given the many type of statement and you have to select the incorrect ones anta kelidane. Illi BT in an BT cotton indicates that the genetically modified crops produced through the biotechnology. ಸೊ ಒಂದು ಬಿಟಿ ಕಾಟನ್ ಅಲ್ಲಿ ಜೀನ್ ಅಥವಾ ಬಿಟಿ ಕಾಟನ್ ಪ್ಲಾಂಟ್ ಗೆ ನಾವು ಬಿಟಿ ಅಂತ ಕರಿತೀವಿ. ಸೊ ದ ಬಿಟಿ ಇಸ್ ಇಂಡಿಕೇಟ್, ಆ ಒಂದು ಬಿಟಿ ಅನ್ನೋದು ಯಾವ್ದಕ್ಕೆ ಇಂಡಿಕೇಟ್ ಕೊಡ್ತಾ ಇದೆ ಅಂತಂದ್ರೆ ಜೆನೆಟಿಕಲಿ ಮಾಡಿಫೈಡ್ ಕ್ರಾಪ್ಸ್ ಪ್ರೊಡ್ಯೂಸ್ಡ್ ಥ್ರೂ ದಿ ಬಯೋಟೆಕ್ನಾಲಜಿ. ಸೊ ಜೆನೆಟಿಕಲಿ ಮಾಡಿಫೈಡ್ ಕ್ರಾಪ್ಸ್ ಬಯೋಟೆಕ್ನಾಲಜಿಯಿಂದ ಮೂವ್ಮೆಂಟ್ ಆಗಿರೋದಕ್ಕೆ ಅಥವಾ ಟ್ರಾನ್ಸ್ಫರ್ ಆಗಿದ್ದಕ್ಕೆ, ಸೊ ದಟ್ ಈಸ್ ವಿ ಕ್ಯಾನ್ ಕನ್ಸಿಡರ್ ದಿ ಸ್ಟೇಟ್ಮೆಂಟ್ ಬಿಟಿ ಅನ್ನೋದು. ಸೊ ದಟ್ ಇದು ಕರೆಕ್ಟ್. ಸ್ಟೇಟ್ಮೆಂಟ್ ಒನ್ ಈಸ್ ಕರೆಕ್ಟ್. ಸೊ ಯು ಹ್ಯಾವ್ ಟು ಫೈಂಡ್ ಔಟ್ ದಿ ಇನ್ಕರೆಕ್ಟ್ ಸ್ಟೇಟ್ಮೆಂಟ್ ಅಂತ ಕೇಳಿದಾನೆ. ಸಾರಿ, ಲೈಕ್ ಇನ್ಕರೆಕ್ಟ್ ಸ್ಟೇಟ್ಮೆಂಟ್ ಅಂತ ಕೇಳಿರೋದು ನಮಗೆ ಜೆನೆಟಿಕಲಿ ಮಾಡಿಫೈಡ್ ಕ್ರಾಪ್ಸ್ ಬಯೋಟೆಕ್ನಾಲಜಿಯಿಂದ ಪ್ರೊಡಕ್ಷನ್ ಥ್ರೂ ದಿ ಅಂತ ಕೇಳಿರೋದಕ್ಕೆ. ಸೊ ರಿಲೇಟೆಡ್ ಟು ದಿ ಬಿಟಿ ಕಾಟನ್ ಇಂಡಿಕೇಟ್ಸ್ ಅಂತ ಕೊಟ್ಟು ಜೆನೆಟಿಕಲಿ ಮಾಡಿಫೈಡ್. ಸೊ ಯು ಹ್ಯಾವ್ ಟು ಫೈಂಡ್ ಔಟ್ ದಿ ಇನ್ಕರೆಕ್ಟ್ ಸ್ಟೇಟ್ಮೆಂಟ್ ನಮಗೆ ಬೇಕಿರೋದು. ಸೊ ಇಲ್ಲಿ ಜೆನೆಟಿಕಲಿ ಮಾಡಿಫೈಡ್ ರಿಲೇಟೆಡ್ ಟು ದಿ ಕ್ರಾಪ್ ಪ್ರೊಡ್ಯೂಸ್ಡ್ ಥ್ರೂ ದಿ ಬಯೋಟೆಕ್ನಾಲಜಿ ಅಂತ ಕೊಟ್ಟಿದ್ದೇನೆ. ಸೊ ಈ ಬಿಟಿ ಕಾಟನ್ ಅನ್ನೋದೇನು ಜೆನೆಟಿಕಲಿ ಮಾಡಿಫೈಡ್ ಕ್ರಾಪ್ಸ್, ಕರೆಕ್ಟ್. ಅಂಡ್ ರಿಲೇಟೆಡ್ ಟು ದಿ ಪ್ರೊಡಕ್ಷನ್, ಸೊ ಇಟ್ ಈಸ್ ಎ ಕಂಪ್ಲೀಟ್ಲಿ ಪ್ರೊಡಕ್ಷನ್ ಆಫ್ ಬಯೋಟೆಕ್ನಾಲಜಿ ಅಂತ ಕೊಟ್ಟಿರೋದು. ಬಟ್ ಬೇಸ್ಡ್ ಆನ್ ದಿ ಬಿಟಿ ಅನ್ನೋದು ಏನಕ್ಕೆ ಇಂಡಿಕೇಶನ್ ಕೊಡ್ತಾ ಇದೀವಿ ಅಂದ್ರೆ ಇಟ್ ಈಸ್ ಎ ಇನ್ಕಾರ್ಪೊರೇಟೆಡ್ ಆಫ್ ಬೆಸಿಲಸ್. ಸೊ ಇಲ್ಲಿ ಏನ್ ಕೊಡ್ತಾ ಇದೀವಪ್ಪ ಅಂದ್ರೆ ನಾವು ಬ್ಯಾಸಿಲಸ್ ತುರಂಜೆನಿಸಿಸ್ ಅಂತಕ್ಕಂಥ ಜೀನ್ ಇಸ್ ವೆರಿ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ಸ್ಟೇಟ್ಮೆಂಟ್ ವೈಸ್ ನೋಡೋದಾದ್ರೆ ನಮಗೆ ಬಿಟಿ ಅನ್ನೋದು ಏನ್ ಇಟ್ ಇಸ್ ಎ ಇಂಡಿಕೇಟಿವ್ ಆಫ್ ಎ ಜೆನೆಟಿಕಲಿ ಮಾಡಿಫೈಡ್ ಕ್ರಾಪ್ಸ್ ಥ್ರೂ ದಿ ಬಯೋಟೆಕ್ನಾಲಜಿ. ಬಟ್ ಬಿಟಿ ಅನ್ನೋದೇನು ಇಟ್ ಇಸ್ ಎ ಮೇಜರ್ಲಿ ದೇರ್ ಇಸ್ ರಿಲೇಟೆಡ್ ಟು ದಿ ಬ್ಯಾಸಿಲಸ್ ತುರಂಜೆನಿಸಿಸ್, ನಾಟ್ ಬಯೋಟೆಕ್ನಾಲಜಿ. ಇಂಡಿಕೇಶನ್ ಮಾತ್ರ ಬಯೋಟೆಕ್ನಾಲಜಿನೇ, ಬಟ್ ಇಟ್ ಈಸ್ ಎ ಟ್ರಾನ್ಸ್ಫಾರ್ಮ್ ಥ್ರೂ ದಿ ಎಲ್ಲಿಂದ ಮಾಡಿಫಿಕೇಶನ್ ಆಗಿರೋದು ಮಾಡಿಫೈಡ್ ಅಂತ ತಗೊಂಡಾಗ ಬಿಟಿ ಅಂತ ಕರಿತೀವಿ. ದಟ್ ಈಸ್ ಬ್ಯಾಸಿಲಸ್ ತುರಂಜೆನಿಸಿಸ್ ಅನ್ನೋದೇ ಇಂಡಿಕೇಶನ್ ಕೊಡ್ಬೇಕು ನಮಗೆ. ಸೊ ದಟ್ ಸ್ಟೇಟ್ಮೆಂಟ್ ನಂಬರ್ ಒನ್ ಅನ್ನೋದು ಇನ್ಕರೆಕ್ಟ್. ಅಂಡ್ ನೆಕ್ಸ್ಟ್ ಬಿ ಆಂಟಿ ಕೊಯಾಗುಲೆಂಟ್ ಅಂತ ಇದೆ. ಹಿರಿಡಿನ್ ಅನ್ನೋದು ಆರ್ ವೆಲ್ ಆಂಟಿ ಕೊಯಾಗುಲೆಂಟ್ ಅಂತಂದ್ರೆ ಒಂದು ಪ್ರೋಟೀನ್ ಆಂಟಿ ಕೊಯಾಗುಲೆಂಟ್ ಆಫ್ ಹಿರಿಡಿನ್. They are being produced from the transgenic of example of Brassicus napusa seeds ಅಂತ ಇದೆ. So iridine is a production from the different organisms. Example of iridineria ಅಂತ ಏನ್ ಕರಿತೀವಿ, ಲೀಚ್ ಇಂದ ನಾವು ಪ್ರೊಡಕ್ಷನ್ ಮಾಡ್ಕೋಬಹುದು. ಬಟ್ ಇಲ್ಲಿ ಎಕ್ಸಾಂಪಲ್ ಆಫ್ ಅನಿಮಲ್ಸ್ ಕೊಟ್ಟಿದ್ದೇನೆ ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ಆರ್ಗನಿಸಮ್ಸ್ ಅಂತ ತಗೊಂಡಾಗ ನಮ್ಗೆ ಸೊ ಆರ್ಗನಿಸಮ್ಸ್ ಅಂತ ಬಂದಾಗ ಸೀಡ್ಸ್ ಆಫ್ ಬ್ರಾಸಿಕಾ ನಪುಸಾ. ಸೊ ಈ ಬ್ರಾಸಿಕಾ ನಪುಸದಲ್ಲಿ ಇರ್ತಕ್ಕಂಥ ಸೀಡ್ಸ್ ಇಂದ ವಿ ಹ್ಯಾವ್ ಟು ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಸಮ್ ಕೈಂಡ್ ಆಫ್ ದ coagulant protein, that is anticoagulant protein ನಾವು ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಮಾಡಬಹುದು. ಸೊ ದಟ್ ಆಪ್ಷನ್ ಅಲ್ಲಿ ಇಟ್ ಈಸ್ ಎ ಕರೆಕ್ಟ್ ಒನ್. ಅಂಡ್ ಸಮ್ ಕೈಂಡ್ ಆಫ್ ಫ್ಲೇವರ್ ಸರ್ವಾ ಅಂತ ಕರಿತೀವಿ. ದ ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ಎಕ್ಸಾಂಪಲ್ ಆಫ್ ಟೊಮೆಟೋಸ್ remain fresh for the longer time than the comparative of normal tomato variety. ಸೊ ಇದು ಮಾಡಿಫೈಡ್ ಟೊಮ್ಯಾಟೋಗೂ ನಾರ್ಮಲ್ ಟೊಮ್ಯಾಟೋಗೆ ಇರ್ತಕ್ಕಂಥ ಡಿಫರೆನ್ಸ್ ಇಷ್ಟೇ, ದೇ ಹ್ಯಾವ್ ಲಾಂಗ್ ಲಾಸ್ಟಿಂಗ್ ಪವರ್, ದಟ್ ಈಸ್ ಎ ಲಾಂಗರ್ ದ್ಯಾನ್ ದ ಫ್ರೆಶ್ ಫಾರ್ ಲಾಂಗರ್ with respect to the normal comparative study about the tomato. So that statement 3 it is also be correct one. So statement 3 also correct. And golden rice is one of the most important transgenic variety of example of Oryza sativa. So common name rice anta karitivi, scientific name Oryza sativa anta karitivi, and which is a very very rich amount of beta carotene, and it can be very very important for the preventing the night blindness in young people. So ಈ ಗೋಲ್ಡನ್ ರೈಸ್ ಅಂತ ಏನ್ ಕರಿತಾ ಇದೀವಿ ಟ್ರಾನ್ಸ್ಜೆನಿಕ್ ವೆರೈಟಿ, ದಟ್ ಈಸ್ Oryza sativa. They have very very rich amount of beta carotene. ಆ ಬೀಟಾ ಕೆರಟಿನ್ ಅಂತಕ್ಕಂಥದ್ದು ಇಂಪಾರ್ಟೆಂಟ್ ಯಾವ್ದಕ್ಕೆ ಅಂದ್ರೆ ದೇ ಹ್ಯಾವ್ ಟು ಇನ್ವಾಲ್ವ್ಡ್ ಇನ್ ದಿ ಕಲರ್ ಬ್ಲೈಂಡ್ನೆಸ್ ಗೆ ರಿಲೇಟೆಡ್ ಆಗಿರ್ತಕ್ಕಂಥದ್ದು. ಸೊ ಪ್ರೊವೈಡಿಂಗ್ ದಿ ನೈಟ್ ಬ್ಲೈಂಡ್ನೆಸ್ ನ ಪ್ರಿವೆಂಟ್ ಮಾಡ್ತಕ್ಕಂಥದ್ದು. ಸೊ ದಟ್ ಇಟ್ ಈಸ್ ಆಲ್ಸೋ ಬಿ ಕರೆಕ್ಟ್ ಒನ್. ಸೊ ಅಕಾರ್ಡಿಂಗ್ ಟು ದಿ ಆಲ್ ಆಫ್ ದಿ ಫೋರ್ ಸ್ಟೇಟ್ಮೆಂಟ್ ನ ನಾವು ಚೆಕ್ ಮಾಡಿದ್ರೆ ಇನ್ಕರೆಕ್ಟ್ ಒನ್ ಬರೋದು ಆಪ್ಷನ್ ಎ ಮಾತ್ರನೇ. ಸೊ ದಟ್ ಆಪ್ಷನ್ ಎ ಓನ್ಲಿ, ದಟ್ ಈಸ್ ಒನ್ will be the correct option for the question number 10. The next question number 11. Which of the following the risks, some kind of risk will be are associated with a modification of the food in a genetically. ಈಗ ನಾವು ಜೆನೆಟಿಕಲಿ ಮಾಡಿಫೈಡ್ ಫುಡ್ಸ್ ಅಂತ ತಗೊಂಡಾಗ ವಾಟ್ ಇಸ್ ದಿ ರಿಸ್ಕಿ ಕಂಡೀಷನ್ಸ್, ಏನಾಗ್ತಕ್ಕಂಥ ಚಾನ್ಸಸ್ ಇರುತ್ತೆ ಹೈಲಿ ನೇಚರ್ ಅಂತ ನೋಡೋದಾದ್ರೆ ಅಂದ್ರೆ ಆಪ್ಷನ್ ಎ ನೋಡ್ಕೊಳ್ಳಿ. ಟಾಕ್ಸಿಸಿಟಿ ಇನ್ ಆನ್ ಹ್ಯೂಮನ್ ಬೀಯಿಂಗ್ ವಿಲ್ ಬಿ like with a maximum consumption of modified category of the food, that is genetically modified food, can increases the toxicity, chemical or poisonous substance in human beings ಅಂತ ಕೊಟ್ಟಿದ್ದಾನೆ. ಸೊ ಸ್ಟೇಟ್ಮೆಂಟ್ ನಂಬರ್ 1 ಅಲ್ಲಿ. ಸೊ ದಟ್ ಈಸ್ totally incorrect. ಅಲರ್ಜಿಕ್ ರಿಯಾಕ್ಷನ್ಸ್ ಇನ್ ಆನ್ ಹ್ಯೂಮನ್ ಬೀಯಿಂಗ್ಸ್ ಅಂತ ಇದೆ. ಸೊ ಅಲರ್ಜಿಕ್ ರಿಯಾಕ್ಷನ್ಸ್ ಆಲ್ಸೋ ಬಿ ಅಕರ್ಸ್, ಬಟ್ like quantity wise with related to the associated genetically modified ಅಂತ ತಗೊಂಡಾಗ very risky ಇರ್ತಕ್ಕಂಥದ್ದಲ್ಲ. ಸೊ ಅಲರ್ಜಿಕ್ ರಿಯಾಕ್ಷನ್ಸ್ ಕೂಡ. And antibiotic resistance in a microorganisms that present an element, it is also be right. And because of they have to death of a maximum number of microorganisms which are already present on the elementary canal gut reason. So that it is also correct in the point of high risky reason. So it is as a minimum quantity. So all of the three will be the correct one because of their toxicity will be slightly increases. Go on, if you take an example for the genetically modified consumption, allergic reaction also increases day by day, and the death of all the type of organisms in an elementary canal also. So all the above will be the a slow process and highly risky conditions related to the overconsumption and modified nature of a genetically organism. That is all the above will be the correct. So ಎಲ್ಲದು correct ಇದೆ. High risky ಅಂತ ಬಂದಾಗ all of the above. So ಎಲ್ಲವೂ minimum quantity ಅಲ್ಲಿ they have to give a risk to the human being. Okay, so that option ಅಲ್ಲಿ D will be the correct. Next question number 12. Read the given statement and you have to select the correct option. Statement number 1. The PCR. We are well on the PCR word indicate for the polymerase chain technique. And it is very useful, or they have to helpful in the detecting the bacterial and viral disease. Even when there is a requirement of some kind of symptoms of the disease are not visible. ಸೊ ಒಂದು ಪಿ ಸಿ ಆರ್ ಟೆಕ್ನಿಕ್ ನಾವು ಯಾವ್ದಕ್ಕೆ ಯೂಸ್ ಮಾಡಬಹುದು ಟು ಫೈಂಡ್ ಔಟ್ ದಿ ಡಿಫರೆಂಟ್ ಟೈಪ್ಸ್ ಆಫ್ ಡಿಸೀಸ್. ಹೌ ವಿ ಹ್ಯಾವ್ ಟು ಫೈಂಡ್ ಔಟ್ ವಿತ್ ಎಕ್ಸ್ಟ್ರಾಕ್ಷನ್ ಆಫ್ ದ ಡಿ ಎನ್ ಎ, multiplication of DNA and insertion of DNA probes and identification of a mutated part, and easily we can identify the disease, that is a viral disease, without availability of any kind of visible symptoms. Okay, so it is a correct option. So option statement number 1, it is a true. And statement number 2 ನೋಡ್ಕೊಳ್ಳಿ, they have production of very low concentration of the bacteria, and the viruses in case of a human body can be detected by amplification of their unique, like example of nucleic acids using the PCR technology. So ಇಲ್ಲಿ if you take an example of symptoms ಅಂತ ಬಂದಾಗ the concentration of viruses in a human being, they are maximum. ಬಟ್ ಇಲ್ಲಿ ಏನಾದ್ರೂ ಲೋ ಕ್ವಾಂಟಿಟಿ ಪ್ರೊಡಕ್ಷನ್ ಆಗಿದೆ ಅಂತಂದ್ರೆ ವಿ ಕ್ಯಾನ್ ಗೋ ಫಾರ್ ದಿ ಪಿ ಸಿ ಆರ್ ಅಂಡ್ ಆಪ್ಲಿಫಿಕೇಶನ್ ಆಫ್ ಜೀನ್ ಅಂಡ್ ವಿ ಹ್ಯಾವ್ ಟು ಫೈಂಡ್ ಔಟ್ ದಿ ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ಪ್ರಾಪರ್ಟೀಸ್ ನ ಚೆಕ್ ಮಾಡಿದಾಗ ವಿ ಹ್ಯಾವ್ ಟು ಐಡೆಂಟಿಫೈ ಈಸಿಲಿ ಡಿಸೀಸ್ ಯಾವುದು, ಬ್ಯಾಕ್ಟೀರಿಯಾನೋ ಆರ್ ವೈರಸ್ ಆರ್ ಎನಿ ಕೈಂಡ್ ಆಫ್ ಅಬ್ನಾರ್ಮಲ್ ಎಫೆಕ್ಟ್ ಅಂತ ವಿ ಹ್ಯಾವ್ ಟು ಈಸಿಲಿ ಫೈಂಡ್ ಔಟ್. ಸೊ ದಟ್ ಎರಡು ಸ್ಟೇಟ್ಮೆಂಟ್ ಕರೆಕ್ಟ್ ಇದೆ ವಿತ್ ರಿಲೇಟೆಡ್ ಟು ದಿ ಎಕ್ಸ್ಪ್ಲನೇಷನ್ ಆಫ್ ಪಾಯಿಂಟ್ ಆಫ್ ವ್ಯೂ ಆಲ್ಸೋ. ಸೊ ದಟ್ ಬೋತ್ ದಿ ಸ್ಟೇಟ್ಮೆಂಟ್ ವಿಲ್ ಬಿ ಟ್ರೂ and 2 is a correct explanation for the statement 1. Option A will be the correct one. Okay. Next question number 13. DNA fingerprinting refers to. So we are well on the mechanism of DNA fingerprinting. The meaning of DNA fingerprinting, identification of dissimilarities in an individual. ನೀವು ಯಾವುದೇ ಒಂದು ಇಂಡಿವಿಜುವಲ್ಸ್ ತಗೊಂಡ್ರೆ we have to find out the some kind of similarities ಏನು, dissimilarities ಏನು ಅಂತ find out ಮಾಡ್ಬೇಕು. So according to the dissimilarities ಅಂತ view ಅಂತ ಬಂದಾಗ we can go for the DNA fingerprinting ಅಂತ ಕರಿತೀವಿ. But this DNA fingerprinting, it is a basically related to the, so ಯಾವ ಪಾಯಿಂಟ್ ಅಲ್ಲಿ ನಾವು ಸ್ಟಡಿ ಮಾಡ್ತಾ ಇದ್ದೀವಿ DNA fingerprinting ಅನ್ನೋದನ್ನ ಅಂತಂದ್ರೆ it is completely related to the molecular analysis of some kind of profiles of a DNA sample. ನಾವು ಡೈರೆಕ್ಟ್ ಆಗಿ DNA ನ ಮ್ಯಾಚ್ ಮಾಡಿ DNA will be present and absent ಅಂತ ನಾವು ಫೈಂಡ್ ಔಟ್ ಮಾಡಲ್ಲ. We have to go for the identification of a gene with a individual differences in the molecular level. Based on the base pair levels, we have to check the sample. So that it is a statement number 1 will be the correct. The reference to the DNA fingerprinting anta tagondre, they are molecular analysis and we have to find out the sample with some kind of profiles of an individual. And B ನೋಡ್ಕೊಳ್ಳಿ, analysis of DNA sample using the imprinting devices ಅಂತ ಇದೆ. Imprinting devices ಇಂದ ನಾವು ಐಡೆಂಟಿಫಿಕೇಶನ್ ಮಾಡಲ್ಲ. And the technique used for the chemical analysis and different specimens in an DNA specimens ಯಾವುದು ತಗೊಳ್ತಾ ಇಲ್ಲ. Technique used for the identification of fingerprintings of an individuals ಅಂತ ಇದೆ. So that both these statements totally wrong. But option A will be the correct one. DNA fingerprinting refers to the molecular analysis of the profile of a DNA sample only. Okay. So that option A will be the correct one for the question number 30. And we're coming to the question number 14. Which of the following statement are correct regarding the process of RNA interference? So one of the most important technique, that is RNA interference, we can go for the resistance power toward the different variety of organisms with a artificially insertion of the double stranded RNA into the organism or a plant, and it can be totally prevent the growth and formation of a protein or the expression of a gene of mRNA. Okay, due to the pairing of complementary toward the RNA produced from the viral or any kind of a nematode. And ಇಲ್ಲಿ the pest resistance properties of plants ಅಂತ ಬಂದಾಗ we can go for the one of the RNA interference ಅಂತ ಕರಿತೀವಿ. RNA interference ಅಂದ್ರೆ meaning ಇಷ್ಟೇ, the expression of RNA na we have to prevent. So that prevention of technique ಏನಂತ ಕರಿತೀವಿ ನಾವು RNA interference technique ಅಂತ ಕರಿತೀವಿ. Okay, and this is used to prevent the some kind of infestation of protozoa. So in the RNA like mechanism ಅಂತ ತಗೊಂಡಾಗ ಸ್ಟೇಟ್ಮೆಂಟ್ ರಿಗಾರ್ಡಿಂಗ್ ದಿ ಅಂತ ತಗೊಂಡ್ರೆ ಲೈಕ್ ಇಟ್ ಈಸ್ ಟೋಟಲಿ ರಿಲೇಟೆಡ್ ಟು ದಿ ಏನಂತ ಹೇಳ್ತಾ ಇದ್ದಾರೆ, ಕರೆಕ್ಟ್ ಸ್ಟೇಟ್ಮೆಂಟ್ ಇನ್ಫೆಸ್ಟೇಷನ್ ಆಫ್ ಪ್ರೋಟೋಸೋವಾ ಇನ್ಫೆಕ್ಟ್ ಆಗಿದೆ ಅಥವಾ ಎನಿ ಕೈಂಡ್ ಆಫ್ ಮಾಡಿಫಿಕೇಶನ್ಸ್ ಇದೆ ಅಂತ ನೋಡಲ್ಲ, because of it is other category and they have to related to the pest resistance. And second one ನೋಡ್ಕೊಳ್ಳಿ ನಮಗೆ, and it is takes place in a some kind of eukaryotic and all the prokaryotic organism as a method of cellular defense. It is a cellular defense. Cells in the production ಆಗ್ತಾ ಇಲ್ಲ, protein defense mechanism will not conducted, so that it is totally not for the cellular defense system. Okay, and see the method which is involved in the silencing of specific mRNA due to the complementary dsDNA. So it is one of the important mechanism, and the specific type of mRNA due to the complementary dsRNA of a molecule. So that statement number 3, it is definitely true, and it is a novel strategy for the production of pest resistance, also the correct one. So only 3 and 4 will be the correct one. The options ಅಲ್ಲಿ ನೋಡೋದಾದ್ರೆ ನಮಗೆ option A will be the correct one for the question number 14. Okay, the next question number 15. The genetic engineering is a very possible because of why there is a possibility of genetic engineering. What is the reason behind that? ಏನಿದೆ ಈ ಜೆನೆಟಿಕ್ ಇಂಜಿನಿಯರಿಂಗು ಅಲಾಂಗ್ ವಿತ್ ಆರ್ ಡಿ ಎನ್ ಎ ಅನ್ನೋದು ಏನಾದ್ರು ಇಂಪಾರ್ಟೆಂಟ್ ಆಗಿ ದೇ ಆರ್ ಪ್ರಿಪೇರ್ಡ್ ಅಂಡ್ ದೇ ಹ್ಯಾವ್ ಟು ಗೋ ಫಾರ್ ದಿ ಡಿಫರೆಂಟ್ ಪ್ರೊಡಕ್ಷನ್ ಆಫ್ ಪ್ರಾಡಕ್ಟ್ ಅಂತ ಬಂದಾಗ ಇಟ್ ಈಸ್ ಅ ಪಾಸಿಬಿಲಿಟಿ. ಬಿಕಾಸ್ ವಾಟ್ ಈಸ್ ದ ರೀಸನ್ ಬಿಹೈಂಡ್ ದಟ್ ಪಾಸಿಬಿಲಿಟಿ, ಏನಿದೆ ರೀಸನ್ ಅಂತ ತಗೊಂಡ್ರೆ we can cut the DNA at a specific site using a restrictions of protozoan. So ಇಲ್ಲಿ genetic engineering is a possible because of ಅಂತ ಕೊಟ್ಟಿದ್ದಾನೆ ವರ್ಡ್. ಸೊ ಯಾಕೆ ಅನ್ನೋದಕ್ಕೆ ಪಾಸಿಬಿಲಿಟಿ ಅಂತ ನೋಡೋದಾದ್ರೆ ಯು ಹ್ಯಾವ್ ಟು ಯು ಕ್ಯಾನ್ ಕಟ್ ದಿ DNA at a specific site by the restriction of some kind of restriction of protozoans ಅಂತ ಕೊಟ್ಟಿದ್ದಾನೆ one point ಅಲ್ಲಿ. And if you take a second one, the restriction endonuclease, that is a enzymes, they are purified from the viruses, can be used in the bacteria. So ಇಲ್ಲಿ purification ಯಾವುದಿದೆ, virus ಇದೆ. And the phenomena, that is a transduction of bacteria, is a well understood ಅಂತ ಕೊಟ್ಟಿದ್ದೀವಿ. And the next one, we can see the DNA by electronic microscope ಅಂತ ಕೊಟ್ಟಿದ್ದೀವಿ. So but according to the statements ಅಂತ ತಗೊಂಡಾಗ ನಮಗೆ the genetic engineering is a very possible because of ಏನ್ ರೀಸನ್ ಇದೆ ಅಂತಂದ್ರೆ we can cut the DNA at a specific site true by the restriction of protozoans ಅಂತ ಇದೆ, it is a wrong. By the restriction of that is a used for the enzymes that is a bacteria. That is a correct. And restriction enzymes are the like example of endonuclease. They are purified from the virus. Totally wrong. They are always purified from the bacteria and it is used for the bacteria only. The phenomena for the transduction of bacteria as well understood. Transduction, meaning it is a possible because of the foreign DNA is inserted into the bacterial DNA incorporation with a carrier of plasmid, cutting of restriction endonuclease and site of a restriction enzymes, all they will be present and the bacteria will be well understood so that it is a correct and we can see the DNA by the electronic microscope. The use of DNA, that is a formation of DNA, we have to visualize in an electronic microscope. But the genetic engineering is nothing but it is a possibility because of carrier of bacteria transduction process of transformation of DNA will be occurs. So that well-known good example will be option C will be the correct one. Okay, out of the four, the C will be the correct option. Next, which of the following is a gene where there is a inserted or they will be introduced to the cotton to protect them from the different types of bollworms. So they are given the bollworms on the so gene of interest ಇದೆ and what kind of the genes which are inserted to protect them from the cotton bollworms ಅಂತ ತಗೊಂಡ್ರೆ they are given the cry1 sorry cry AC cry AB and BTAC and BTAB. There is no BTAC and BTAB ಅನ್ನೋ ವರ್ಡ್ ಯೂಸ್ ಮಾಡಲ್ಲ ನಾವು and NIF genes ಅಂತ ಕರಿತೀವಿ and there is a presence of EB and AC will be available. But according to the statement, like ballworms basically related to the cry1 AC with respect to the character AB, they are controlling power for the ballworms. Another so option B, C will be the correct one for the question number 60. Next, the BT protein that is a completely crystals present in an bacteria like example of Bacillus thuringiensis and do not kill the bacteria themselves. Already first questions ಅಲ್ಲಿ ನಾವು study ಮಾಡಿದೀವಿ. The Bacillus thuringiensis production of protein of the crystalline substance ಏನಿದೆ it cannot kill the themselves ಅಂತ ಇದೆ because of they are inactive form. So ಯಾಕಿರುತ್ತೆ reason ಇಷ್ಟೇ they are inactive formation ಅಷ್ಟೇ and the bacteria are resistance to the toxin bacteria resistance ಇದೆನಾ ಅಂತ ಕೇಳಿದಾನೆ so there is no any kind of resistance to the toxin toward the bacteria so that option A wrong. B, the toxin occurs as an inactive and protoxin in an bacteria. The toxin occurrence ಏನಿದೆಯಲ್ಲ totally they are inactive protoxin and in an bacterium ಅಂತ ಕೊಟ್ಟಿದ್ದಾನೆ. So inactive protoxin it is a correct one with related to the bacterial strain ಅನ್ನೋದು so formation of the inactive toxin that is we can call the protoxin ಅಂತ ಕರಿತೀವಿ so that they have not kill the bacteria themselves. There is a need of alkaline pH for the conversion of crystalline into soluble form. Crystalline the soluble formation ಆಗ್ಬೇಕಂದ್ರೆ there is a need of alkaline media so that optionally be correct. Bacteria encloses the toxin in a special sacs ಅಂತ ಇದೆ and the toxin ನ ಸ್ಟೋರ್ ಮಾಡ್ತಕ್ಕಂಥ sacs will not present none of the above ಬರಲ್ಲ so option B will be the correct one for the question number 17. And 18, what is the permanent cure for the adenosine deaminase? Already we are well known about the gene therapy. The modification or a replacement of a defective gene, manipulation of a defective gene and insertion of normal gene, that is a ADA deficiency in an example of children. And there is a bone marrow transplantation ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ ಇಲ್ಲಿ completely or a permanent cure method now we have to find out. Bone marrow transplantation in the we have to cure but permanent cure ಆಗಲ್ಲ. Always we have to insert the new kind of bone marrow transplantation so that it is not a particularly permanent cure. And the enzyme replacement therapy which is a functional ADA gene is given to the patients by the injection. So now ADA functional gene ನಾವು artificially insert ಮಾಡ್ಬೋದು but when we are inserted the ADA then the person they have to calibrate the production of immunity. After the expression of the gene there is again no production of protein or no any kind of immunity so that it is also not a permanent method for the human being. and transmission of genetically engineering lymphocytes in which the functional ADA and CDA introduced into the patients ಅಂತ ಇದೆ. So it is also one of the important near to the permanent cure, but artificially we have to insert repeatedly we have to insert the lymphocyte for the particular that type of a person. Some kind of the insertion will be some kind of antibody will be the given with a particular time interval so that it is also not a permanent cure for the ADA deficiency. So that option C also be incorrect. And there is introduction of gene isolated from the bone marrow cells which is produced in ADA into the cells of patient at an early embryo stage. Now, in other words, if you have detect the ADA deficiency in early embryo and the bone marrow transplantation could be occurs in an early embryonic condition stage, in that time duration only we have to get a permanent cure to the ADA deficiency. So that option, the definitely D will be the permanent cure method for the syndrome of deficiency disorder of ADA genes. Correct. Okay. And the next one, question number 19. The golden rice developed through the transgene approach and is a very, very enrichment with that. ಈ ಗೋಲ್ಡನ್ ರೈಸ್ ಅಂತ ಕರಿತೀವಿ. So we are well on the golden rice because of they are made up of beta carotene, and these beta carotene it is a increases the major important content. All example highly lysines ಅಂತ ಕೊಟ್ಟಿದ್ದೇನೆ, glutamines ಅಂತ ಇದೆ, vitamins ಅಂತ ಇದೆ, and methionines ಅಂತ ಇದೆ. So the golden rice have beta carotene and it is a very very high rich amount of vitamin A content will be present and it can be used for the treat regarding the prevention of a night blindness disease. So that option D will be the correct for the question number golden rice with an enrichment with vitamin A. Okay. And the next question number 20, JEAC ಅಂತ ಇದೆ. Example of GEAC is indicate for the ಏನಕ್ಕೆ ಇಂಡಿಕೇಶನ್ ಕೊಡ್ತಾ ಇದೀವಿ ಅಂದ್ರೆ ಅಪ್ರೂವಲ್ ಕಮಿಟಿ ಅಂತ ಕರಿತೀವಿ. So genetically engineering approval committee will be the correct. Expand ಅಂತ ಕೇಳಿದಾಗ it can stands for ಏನಕ್ಕೆ ಬರುತ್ತದು the genetically modified or the genetic engineering approval committee. They have to approve the research field and they have to approve the marketing field of any kind of new product which is comes from the research or engineering technique of genetic. So it is a very very important like example of the committee, that is genetic engineering approval committee. So these are the some questions of the biotechnology applications and its the process and principle regarding the applications. And in the next class, we can continue with the concept of these applications only. Okay, thank you.